Hello, welcome to Direct Stars, and you're welcome to our biology class today. So, in our class today, we are going to be talking about microscopy, and our subtopic for today's lesson is microscope. So, before we go further, our objectives for today's lessons are at the end of the lesson, you should be able to understand the principles of microscopy and how it works. Secondly, you should be able to mention the different parts of a microscope, and also you should be able to explain the functions of each part of the microscope. So, when we talk about microscopy, what are we talking about? Microscopy is a technical field that is involved with the use of microscopes to view samples and objects that cannot be seen with the unaided eye. So please don't make a mistake in using the word microscopy and microscope. So this is what we mean when we talk about microscopy. But when we talk about microscopes, what are we talking about? Microscopes are optical instruments that are used for viewing very small objects, such as mineral samples or animal or plant cells. Typically magnify these images or these objects several hundred of times. So this is what we mean when we talk about microscopes. We are simply talking about the instruments. But when we talk about microscopy, we are talking about the field that is involved with the use of these microscopes. So let's look at a brief history of the microscope. The invention of the microscopes started in the 1950s by makers of eyeglasses. 75 years later, an English scientist named Robert Hooke invented the microscope, in which he was able to discover the cell in the year 1665. And because of his findings, he is also called the father of cells. But we are not talking about the cells in this class. We are focusing on microscope. So when Robert Hooke invented his microscope, it didn't quite look like this particular microscope we are seeing nowadays. It was quite different. Okay, so he invented the microscope, and with the aid of his microscope, he was able to find out a few things that most scientists didn't know of as at that time. So with his microscope, he was able to view the cell. He looked at the cells of a tree, and yeah, and they looked exactly just like a honeycomb. You know those tiny little compartments you see in a honeycomb, the way it is arranged, that was exactly what he saw with the aid of his microscope. And this led him to writing the book which he called the Micrographia. Here he filled it up with different diagrams and images of cells and all that. Okay, I'm sure now you know what we're talking about when we're talking about the microscope. So now let's take a closer look at this microscope. If you take a look at this diagram, you will see that there are some labelings on it. Yeah, so now let's look at these labelings. We have the eyepiece, we have the arm, we have the nose piece, the objective lens, the stage, the diaphragm, coarse adjustment, fine adjustment, light source, and the base. So let's talk about a few of them. So now we have the eyepiece. What is the function of the eyepiece? The eyepiece is simply is the part where you look through. When you're making use of a microscope, the eyepiece is where you put your eyes on to look through it. So this is often magnified to like 10 times. The next is the objective lens. Usually there are three to choose from. As you can see in this diagram, there are three different lenses to choose from, okay? So the smallest will be the easiest to focus. So we have the times 5, times 10, and times 25. So it is ideal that you start off firstly with the times 5. So after you've been able to focus this lens on your specimen or object, then you can adjust it or change it to whichever other magnification or lens that you want to use, okay? Then next comes the stage. The stage, when we talk about the stage, this is it right here. The stage, what is it used for? It's used to hold the sample securely, often using two metal clips. If you take a look at this diagram here, you can see, you can find the two metal clips right on the stage. Next comes the specimen. So you can here you can choose to use any specimen that you want to uh, find out more about. It could be an organism, it could be an object, whatever it is. So here you put here is where the specimen comes in. So your specimen is usually placed in a drop of water or stain on a microscope slide under a very thin glass cover slip. So in your microscope, you're going to find this thin glass cover. So for you to view any object or any specimen, you have to place it, you have to place that specimen in a drop of water or a stain on a microscope slide, and then you cover it with a thin glass cover slip for you to be able to view what you want to view. So after the specimen, next comes the mirror. Where is the mirror in your microscope? This is it right here. Okay, this mirror could be replaced with a light source in other advanced microscopes such as uh, other advanced microscope basically. It could be replaced with a light source, and some may just have a mirror. This mirror is used to like reflect the light that is coming from the microscope so that you can be able to see clearly what you're finding more on about. So after the mirror, next comes the coarse focus. So the coarse focus or adjustment, this is it right here. So what is it used for? This quickly and easily moves the stage up and down. So it's used to help move the stage quickly up and down, while the fine focus is used for moving the stage also slowly. Okay, that is just what it is used for. Okay, so having said this, I'm sure you know what a microscope is right now. So let me ask you, what is a microscope? Good. A microscope is an optical instrument that is used for viewing very small objects, such as mineral samples or animal cells or plant cells or anything whatsoever that you want to view. Good. So that will be all for our class for today. I hope you enjoyed the class. Thank you for watching.